Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine your brain for a second. It's this incredibly intricate network, right? And every single cell is like a tiny, bustling office inside. And in each office, you've got the central command center, the nucleus, holding the master blueprint, your deoxyribonucleic acid, your DNA.
That's right, the core instructions.
Exactly. And from this blueprint, these crucial instructions get sent out as genetic messages. They travel to other parts of the cell to get all the vital work done.
A constant flow of information.
But what happens if that internal mail system, the one delivering these super precise instructions, suddenly goes, well, completely off course?
Things can go wrong, definitely.
Today, we're taking a deep dive into some really groundbreaking new research. It seems to uncover a fundamental, shared problem right at the heart of two devastating brain conditions: Alzheimer's disease and frontotemporal dementia.
Yeah, this is fascinating work. We've been looking at a health news article and, maybe more importantly, the detailed scientific paper it's based on.
Hot off the press, basically, from the Crick Institute and University College London.
That's the one.
So our mission today is to really unpack this discovery. We want to understand what's actually happening inside those brain cells when these vital genetic messages get misplaced.
And also look at a potential treatment that's, quite surprisingly, already in clinical trials for other things.
Okay, let's explore this together.
Well, what's really fascinating here starts with something pretty fundamental: how a cell normally handles its genetic messages.
Okay.
Think of messenger ribonucleic acid, that's messenger RNA, as the crucial emails carrying instructions.
Right, the messages themselves.
Exactly. They come from the DNA, which is kept safe in the nucleus, and then they travel out into the cytoplasm, that's the main body of the cell. And out there, those instructions are used to build proteins, which are basically the cell's workers doing all the jobs. It's the core messaging system.
Okay, makes sense, like an internal mail system.
Precisely. And the first clue that something might be, profoundly wrong with this system actually came from earlier research at the Crick Institute.
Oh, okay. What did they find then?
They discovered that some of these messenger RNA molecules were being found in the wrong place inside nerve cells, specifically in cells with genetic mutations known to cause motor neuron disease.
Wow, okay. So not where they were supposed to be.
Not at all. And that unexpected finding was really the spark that ignited this current, much broader investigation into these dementias.
So if this basic messaging system was already showing glitches in motor neuron disease, what did this new deep dive uncover when they specifically looked at Alzheimer's and frontotemporal dementia? Was the same kind of misplacement happening there too?
That was exactly the question. The team's main goal was to figure out if this same kind of messenger RNA misplacement also happened in these two major forms of dementia.
Right.
And to answer that, they used a really insightful approach using human-derived cells. It's quite clever, actually.
How did they do it?
Well, they started by taking skin cells from people who had inherited forms of Alzheimer's disease. So these individuals carried specific mutations in genes we know cause the disease, like amyloid precursor protein or presenilin-1.
Okay, the known genetic links.
Exactly. And they also used skin cells from people with inherited frontotemporal dementia, specifically those with mutations in the velocin-containing protein gene.
Got it.
Then, here's the really cool part. They transformed these skin cells back into a stem cell state, like rewinding their development.
Induced pluripotent stem cells, right.
iPS. Precisely. And from that stem cell state, they carefully guided them to specialize into cortical neurons.
Ah, the actual brain cells affected.
The very type found in the brain's cortex that are hit hard in these conditions. Then, to understand what was different, they carefully compared these diseased neurons to cortical neurons they developed from stem cells taken from healthy donors.
Okay, so a direct comparison.
A direct comparison. And once they had these neurons, the researchers physically separated the cells into their two main compartments.
The nucleus and the cytoplasm you mentioned.
Exactly. The nucleus holding the DNA and the cytoplasm, which is, the rest of the cell. Then they meticulously mapped the exact locations of all the messenger RNA molecules within these two areas.
That sounds incredibly detailed, like tracking every single piece of mail. What did they actually see? Were the messages getting to the right cellular addresses?
Well, in the control neurons, the healthy ones, the messenger RNA molecules were consistently found right where they should be. Everything looked normal. operating as expected.
Okay, the mail was delivered correctly there.
Yes, but, and this is the key finding, this was clearly not the case in the neurons with those neurodegenerative mutations. They found a significant number of messenger RNA molecules somewhere between, say, 82 and 140. Depending on the mutation, we're ending up in different places than they ought to be.
Wow, it's quite a few going astray.
It is, and here's where the common thread really emerged. There were 10 specific messenger RNA molecules that were consistently misplaced across both Alzheimer's disease and frontotemporal dementia models.
10 specific ones, common to both conditions.
Yes. One of these was found wrongly located in the nucleus, while the other 9 ended up incorrectly in the cytoplasm.
Okay.
And critically, all 10 of these commonly misplaced messenger RNA molecules carried instructions from genes directly related to mitochondrial function.
Mitochondria, the cell's power plants.
The very same. The cell's essential energy producers. And it gets more compelling. The researchers also looked at postmortem brain tissue from people who had frontotemporal dementia.
Real human brain tissue.
Yes. And they found that 4 of those 9 messenger RNA molecules that were misplaced in the cytoplasm were significantly associated with worse disease outcomes. They seem to be clear indicators of the disease's progression.
So it really sounds like the mitochondria, the cell's energy factories, are somehow fundamentally caught up in this misdirection.
It strongly points that way.
So given that the messenger RNA related to mitochondrial function was ending up in the wrong place, did the researchers find out anything else about these, vital cellular powerhouses? Were they compromised in other ways?
Yes, absolutely. The problems with the mitochondria actually went even deeper.
Okay.
Since the messenger RNA from mitochondria was consistently misplaced, the team logically looked next at the DNA inside the mitochondria. That's called mitochondrial DNA, or mtDNA.
Right. Mitochondria have their own DNA.
They do. And what they found was substantial amounts of this mitochondrial DNA outside the mitochondria, floating around in the cytoplasm.
Leaking out.
That's exactly what it suggested.
Yeah.
That not only the messenger RNA messages, but also the mitochondrial DNA blueprints themselves were somehow leaking out from these powerhouses.
How could they tell for sure?
They actually confirmed it visually using super-resolution imaging. They could see this abnormal buildup of mitochondrial DNA in the cytoplasm.
Wow. Okay, so leaky mitochondria. What else?
Well, beyond the leakage, they also observed that the diseased neurons just had fewer mitochondria overall.
Fewer power plants.
Exactly. And the ones that were left were smaller in size and, maybe unsurprisingly, produced less energy. So the cell's power supply was basically dwindling.
That sounds bad enough on its own.
It is. But the implications didn't stop there. This leaked mitochondrial DNA floating in the cytoplasm also activated an inflammatory pathway in the brain.
Inflammation? How?
It switched on something called the cyclic GMP-AMP synthase stimulator of interferon gene signaling pathway. It's a bit of a mouthful.
Yeah, definitely. CGS sting pathway, is that sometimes?
Yes, often shortened to CGS sting. It's part of the innate immune response. The researchers believe this inflammatory response, triggered directly by that leaked mitochondrial DNA, could be directly linked to some of the neurodegenerative symptoms we see in these diseases.
Okay, so we have misplaced messages, leaky mitochondria, less energy, and inflammation being triggered. That sounds like a cascade of problems.
It really does paint a picture of significant cellular dysfunction.
So, given these really fundamental issues, did the researchers find anything that could actually, restore some order to this cellular chaos? Is there a way to put the mail back in the right boxes and maybe fix those leaky power plants?
They did look for an intervention. As a crucial part of their investigation, the team treated these diseased cells with a specific drug called ML-240.
ML-240, what does it do?
It specifically inhibits a particular function of that protein we mentioned earlier, the volosin-containing protein, or VCP. VCP is thought to be involved in things like cellular protein recycling and transport.
Okay, so they targeted that protein.
And the results were quite positive, pretty encouraging, actually. Treatment with ML-240 effectively returned those misplaced messenger RNA molecules back to their typical correct locations.
It fixed the mail delivery.
It seemed to, yes.
Yeah.
And its beneficial effects also extended significantly to the mitochondria.
How so?
Well, the drug dramatically reduced the amount of that mitochondrial DNA leakage we talked about. It also prevented those inflammatory pathways like CGS sting from getting activated.
So less inflammation.
Less inflammation trigger, yes. And crucially, it raised mitochondrial activity back towards normal levels. It even helped restore the mitochondrial membrane potential, which is vital for making energy, and it reduced the levels of cyclic GMP, AMP, that key molecule in the inflammatory pathway.
So let me get this straight. A drug targeting this specific protein function, the velocin-containing protein. And you said it's already being looked at in clinical trials for other things.
That's right. It's being explored elsewhere.
And this drug could actually fix these fundamental issues in the cell model, from the misdirected messages to the leaky DNA and the inflammation. That seems like a really significant finding for you, the listener, to consider.
It is indeed. And the researchers themselves offered some important insights into what this could all mean. Christy Hung, one of the lead authors, she noted that they seem to have identified a, quote, common phenomenon across different types of dementia and even motor neuron disease, that these messages carrying instructions from the DNA are ending up in the wrong places, which really messes with how cells function.
A common problem across different diseases.
Right. And she even suggested that the effects on the mitochondria, the leakage of mitochondrial DNA triggering inflammation, she said that may even be a universal mechanism in neurodegeneration.
A universal mechanism. That's a bold statement.
It is, but it reflects the potential significance. And Ricky Patani, another senior group leader on the study, he really emphasized the, quote, desperate and unmet need to understand the fundamental molecular mechanisms behind Alzheimer's and frontotemporal dementia.
Which is absolutely true.
Yeah. And he stated that their findings suggest that this misplacement of, as he put it, genetic building blocks like messenger RNA and mitochondrial DNA could be a common mechanism that is potentially therapeutically tractable.
Meaning something we could actually target with treatments.
Exactly. Therapeutically tractable.
So if we sort of zoom out and connect this to the bigger picture, this study started by looking at pretty classical ideas about how genetic material moves in cells.
Right. Nucleus to cytoplasm transport.
But it led them somewhere much broader. It feels like it shifts our understanding beyond just, say, problems with standard transport pathways.
I think that's a good way to put it. It moves towards a more integrated view of just overall cellular integrity.
Yeah.
It highlights how critical the precise location of genetic information is, both nuclear and mitochondrial, and importantly, how our own innate immune system reacts when things go wrong at that fundamental level.
Okay. Now, despite these really exciting findings, it's always important to talk about limitations, right? What should we keep in mind here?
Absolutely. That's crucial in science. A key point is that this study used an in vitro cellular model, specifically those human-induced pluripotent stem cell-derived cortical neurons.
The lab-grown brain cells.
Right. And while these models are highly relevant for digging into disease mechanisms and they offer really valuable insights, we have to remember they might not fully capture the incredible complexity and the whole environment of how Alzheimer's and frontotemporal dementia actually develop inside a living person.
It's a model, not the real thing.
Exactly. It's a very useful model, but still a model. Also, actually validating this messenger RNA, shifting between the nucleus and cytoplasm in postmortem human brain tissue is, well, it's quite challenging.
Why is that?
Well, brain tissue itself is a mix of many different cell types, neurons, glial cells, all sorts. So that makes it hard to get neuron-specific insights.
It's not just pure neurons you're looking at.
Right. And physically separating just the cortical neurons from human postmortem brains is technically really difficult because they're quite fragile after death.
Okay, so practical challenges in confirming this directly in human brains.
Yes, significant ones.
Understood. So given those considerations, what are the really crucial next steps? Where do we go from these lab findings towards something that might actually help people? And what kind of complexities might researchers run into?
Well, the immediate next step, logically, is to test whether these velocin-containing protein inhibitors like ML240 or similar drugs work effectively in more complex model organisms.
Moving up from cell cultures to, say, animal models.
Precisely. That's the next stage of validation. And it's also absolutely vital to investigate whether this same misplacement of genetic building blocks the messenger RNA and mitochondrial DNA also happens in the sporadic forms of these diseases.
Right, the much more common cases that aren't directly inherited.
Exactly. Most cases of Alzheimer's, for instance, are sporadic. We need to know if this mechanism applies there too.
That's a huge question.
It is. And another really crucial consideration for developing any future drugs based on this involves thinking about tau pathology.
Tau, the protein that forms tangles in Alzheimer's.
That's the one. Tau clumps are a key feature in Alzheimer's and some other neurodegenerative diseases. And research suggests that this velocin-containing protein actually plays a complex role in regulating tau.
Okay, so inhibiting it might affect tau.
It might, and potentially in different ways, depending on how you inhibit it.
Mm-hmm.
There's some evidence suggesting different classes of VCP inhibitors might have divergent effects on how tau spreads. For example, the drug used in this study, ML240, has actually been reported in other studies to potentially increase tau seeding efficiency, basically, help tau clumps form. Whereas another inhibitor, called NMS-873, seemed to reduce tau spreading.
Wow, so opposite effects from different inhibitors targeting the same protein.
It appears so, which really points to a complex and specific role for phallosin-containing protein in managing tau. It underscores the need for real caution and very comprehensive testing in future drug development. You need to make sure a potential treatment doesn't inadvertently make another aspect of the disease worse.
Okay, so it's definitely not straightforward. There are complexities to navigate.
Definitely, as with most things in neurodegeneration.
So wrapping this up, this deep dive has really shown us that even within the incredibly intricate world of our cells, something that sounds almost simple, like a misdirection of genetic mail, those vital messenger RNA molecules can lead to really profound consequences for our brain health, potentially affecting devastating conditions like Alzheimer's disease and frontotemporal dementia.
Yeah, this research truly opens a new, and I'd say tantalizing window into potentially treating these incredibly complex neurodegenerative diseases. The idea of targeting what might be a universal cellular error, it's compelling.
It really is.
The notion that restoring order at such a fundamental, almost invisible level could have such a widespread positive impact on brain function, that's a powerful idea.
So as you, our listener, think about this, maybe consider how much of our understanding of these complex diseases is still very much evolving.
Constantly.
And how a seemingly small detail, like a misplaced genetic message, might actually hide a universal mechanism for illness and perhaps a powerful new pathway towards healing. What new connections or possibilities does this idea spark for you?
It's definitely food for thought.
We certainly hope this deep dive has given you some valuable insights and maybe a deeper appreciation for the cutting-edge science that's happening right now.
Yeah, fascinating stuff.
Until next time, keep learning and stay curious.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.